An important job in cells is to move cellular products around the cell so they can be used by another part of the cell. So for example, proteins that are produced by ribosomes often need to move to other parts of the cell where they can perform their function. Sometimes these products are required for use in other cells, so they have to move from one cell to another. And in addition, wastes need to be removed and transported out of cells. Now these jobs are carried out by specialized organelles that work together to move substances around cells and out of cells. And these are the endoplasmic reticulum, the Golgi apparatus, and lysosomes. I'm going to speak about each of these and their function, as well as explaining how they work together. The endoplasmic reticulum is a network of sacs and tubules composed of membranes. Endoplasmic means within cytoplasm or within the cytoplasm, and reticulum means network. So it's basically referring to the network that exists within the cytoplasm, that is the endoplasmic reticulum. The endoplasmic reticulum originates at the nuclear envelope and winds throughout the cell. Close to the nucleus, the endoplasmic reticulum is studded with ribosomes. And these ribosomes make proteins that enter the inner compartment of the endoplasmic reticulum. These proteins are destined to be secreted from the cell. Therefore, the ER is actually an intercellular transport system, transporting proteins from one cell to another. And of course, the rough ER gets its name from the ribosomes that give these membranes a roughened appearance. Now, adjacent to the rough ER is a section of the network called the smooth ER, or the smooth endoplasmic reticulum. And the function of the smooth ER depends on the type of cell it's located in. Its main role is to transport proteins. However, it can also synthesize or make lipids, such as those that will end up in milk, and it also assists the manufacture of the plasma membrane. Now in liver cells, the smooth endoplasmic reticulum houses enzymes that detoxify drugs and poisons. And in muscle cells, a specialized type of smooth ER restores and delivers the calcium ions required for muscle contraction. So this is a pretty important organelle, the smooth and rough ER. This image shows an illustrated version of smooth and rough ER, as well as an electron micrograph of rough ER. And as you can see from this image up here, the rough ER originates at the nuclear envelope. This is the rough ER. And you can tell that because it's studded with ribosomes. And as I mentioned earlier, those ribosomes are producing proteins that will go into the rough ER, and those proteins are destined for transport to other cells. Now, ER that does not have ribosomes on it is called Smoothie Er. So again, in a stylized version, here is the rough ER. These are the ribosomes from which the rough ER gets its name. And this is the smooth ER, which does not have any ribosomes on it. Now, the lipids and proteins made by the ER exit the organelle in something called vesicles, and these are vesicles here. So that's a vesicle. And a vesicle is basically a transport system, and it pinches off from the tubular endings of the ER membrane and takes its contents to the next stop in the production line, which happens to be the Golgi apparatus. So the Golgi apparatus is a flat membrane-enclosed sac that functions as a protein processing center. So it processes proteins. In fact, proteins from the endoplasmic reticulum pass through a series of Golgi sacs where they complete their intricate folding and become functional. So you may remember, if you think back to the biomacromolecules, that proteins need to actually form particular shapes by folding into secondary, tertiary, or quaternary structures. And it's that structure, or that folding, that shape, that gives the protein its function. And that's one of the jobs of the Golgi apparatus. Enzymes in the Golgi apparatus also manufacture and attach carbohydrates to proteins or lipids, forming glycoproteins or glycolipids. The Golgi apparatus sorts and packages materials into vesicles, which move towards the cell membrane. Some of the proteins it receives from the ER will become surface proteins on the surface of the cell membrane, and others, such as milk protein and fat, are packaged for secretion out of the cell. So here is an illustration of a Golgi apparatus, as well as an electron micrograph image of a Golgi apparatus right here. You'll often hear Golgi body. It's the same organelle, it's just with a slightly different name. So we know that the Golgi apparatus receives vesicles, which contain proteins. So these are vesicles, and these vesicles are actually entering the Golgi apparatus. Once they enter the Golgi apparatus, the proteins that are contained inside the vesicles complete their folding process so that they're fully functional proteins. Now on the other side, you end up with vesicles that have been processed so that they actually have complete functioning proteins inside of them. So these vesicles are leaving. And so here's another example of a vesicle in the electron micrograph, which you can see quite clearly having pinched off the end of the membrane of a Golgi apparatus.